Все, могу говорить. Сейчас, да? Спасибо. Dear students, tell me please, write down the answer in the chat. Are you coming for lecture for microbiology or for the other discipline in the tutorial number 7? Because we have problems now with identifying of the rooms for lectures. Answer please in the written form. What the lecture do we have now in the auditorium number 7? Is it microbiology or different? If we have lecture for microbiology, topic of today's lecture is the human retroviruses, HIV and AIDS. I will start to read the lecture and wait for your answer, for your comment. What the discipline do we have? Human retroviruses, this is a large number of different viruses. One of them is known as HIV, human immunodeficiency virus, that causes AIDS. AIDS, this is acquired immunodeficiency syndrome, very severe disease, causing complications in the form of the so-called opportunistic infections. Opportunistic infections, what in the slide here you see in the low part of the slide, photograph of the skin of the AIDS patient that developed the sporotrichosis. This is a infectious agent that usually causes infection in only immunosuppressed people who have low level of the immune response. Commonly, retroviruses, including human retroviruses, are RNA-dependent DNA polymerase contained viruses. They have their envelope viruses having positive sense ribonucleic acid genome, and they have unique morphology. In the photograph, in the upper part of the slide, you see typical HIV viruses, virions that have the cylindrical core, central part of the virus that makes virus look like the pupil or eye of the cat. Typical distinctive property of all the retroviruses is the presence of the variants of the RNA-dependent DNA polymerase. RNA-dependent DNA polymerase is encoded by the virus genome. Also, this enzyme is named as the reverse transcriptase. Reverse transcriptase replicates through a DNA intermediate. DNA intermediate, this is a DNA copy of the genomic ribonucleic acid of the virus genome. The severe disease, most famous, caused by human immunodeficiency virus, is the AIDS. Ladies and gentlemen, I don't see your presence here. Are you here? I will read the question to you. Dear guys, I have a question to you. Do you have lecture in the for microbiology now in the auditorium number 7. If you have, answer me, please, in the written form. I wait for your answers, for your presence in this lecture. Because, unfortunately, our administration organized the jam. And me was informed that auditorium for lecture is number 30. Really, after some people thought that 7. But me now try to read the lecture in the auditorium number 7. Are you present? If present, answer me, please. Really, do you have lecture for microbiology number now is the auditorium number 7? Answer me, please. Waiting for your answer, I will try to continue the lecture. Ответить. Вот. Как теперь надо. Это вроде приходил, уходил один человек.
Один человек. Что-то так сами звонили по поводу 7-й аудитории вашей.
Что?
Ну, по поводу 7-й аудитории, потому что у вас только немного студентов.
Да, видимо, был один человек, такое впечатление, что он не в курсе об этом. И на письмо пока никто не ответил.
Ну, сейчас я позвоню.
Спасибо. Ну, if some of you here, I will continue. Global HIV statistic. This is statistic of the human immunodeficiency virus infection from the beginning of the pandemic caused by this virus. 75.7 million people have become infected with HIV since time of the discovery of this virus, since 1983 till 2019. More than 35 million people. 32 million people died from AIDS during this period. In the 2020, approximately 37.6 million people globally were living with HIV. That means half of the infected people during all the pandemic was killed, about half survived. In 2019, about 2 million people become newly infected with the disease. In 2020, around 700,000 people died from AIDS latent illness worldwide. But this is a characteristic of the importance of this virus for humankind now. Human retroviruses are the large group of the different viruses. All they are members of the Retroviridae family. These Retroviridae families are divided to many subfamilies. They are shown by the blue or dark blue squares here. Human retroviruses are here shown in the subfamily that is named delta retroviruses, so-called lentiviruses, the next subfamily, and spumaviruses. Here you see 3 subfamilies, but really 4 subfamilies containing these human retroviruses. 4 subfamilies that are identified now as the human retroviruses. It means retroviruses that may occur in the human body and replicate in the human body are subfamilies: spumavirinae, oncoronaviridae, lentivirinae, and endogenous viruses. First subfamily, spumavirinae. Spumavirinae contains human formiviruses. The next subfamily is oncornaviruses. This word consists of different parts. Onco means tumor generating RNA. This is a ribonucleic acid genome of the viruses. Acarnaviruses consist of human T lymphotropic virus type 1, HTLV1, human T lymphotropic virus type 2, and human T lymphotropic virus type 5. T lymphotropic means that oncoviruses cause tumor transformation of the T lymphocytes. They are host cells, T lymphocytes for oncoviruses. The next subfamily, lentivirinae. Lentivirinae contains human immunodeficiency virus type 1 and human immunodeficiency virus type 2. There are two species of human immunodeficiency viruses causing AIDS. And last subfamily, endogenous viruses or human placental viruses. Human placental viruses. Brief characteristic of the viruses associated to certain subfamilies. Subfamily spumavirinae. These viruses, spumavirinae viruses, cause no clinical diseases known now, but they reside in the virus-infected cells. They produce characteristic vacuolated, foamy, cytopathological effect. So-called human formivirus causes cytopathology effect of the monocytes. Second subfamily, endogenous viruses. These viruses have retroviruses sequences that are integrated into a genome. These viruses may take up to 1% of chromosome material of the human cells. They are ultimate parasites, transmitted vertically. Vertically, it means from the mother to the baby. Variants of these viruses are unknown. These viruses have been discovered as the genetic addict to the human genome. These indigenous viruses also are not associated with the non-human pathology. In the human pathology, important role is the role of the oncornavirinae or oncornaviruses subfamily. But in the illustration, you see 4 squares, and the lower left shows the so-called C-type retrovirus structure. With C-type retrovirus structure with the icosahedral capsid contains genome inside and spherical shape of the virus. This is typical for oncornavirinae, so-called C-type of the human retroviruses. Oncornavirinae are cancer-causing retroviruses. Members of this group are human T lymphotropic virus type 1, human T lymphotropic virus type 2, and type 5. They are called RNA tumor viruses also. These oncornaviruses immortalize or transform target cells. Many of these viruses express viral pathogenesis that is analogous of the cellular growth controlling genes of the human cells. And finally, major topic of our today's lecture is lentivirinae. Lentivirina family consists of the human immunodeficiency virus 1 and human immunodeficiency virus 2, HIV1 and HIV2. Peculiarity of this virus is different from the coronaviruses. Lentivirina are non-oncogenic viruses. They don't form tumor transformation of cells. In the contrast, they establish cytocidal effect. It means they are killers of the infected cells. They're slow viruses that slowly replicate in the human cells. They may cause neurological symptoms and immunosuppressive disease. Usually, lentivirina, and this is a characteristic of the AIDS, they cause slow, progressive chronic diseases, including AIDS. Acquired immunodeficiency syndrome, this is a disease caused by HIV viruses, was primarily discovered in the early 1980s, about 1983. And this disease has been described as the disease of the homosexual man, Haytons, heroin addicts, and hemophiliacs. So-called 4H club, H, homosexual men, H, Haytons, H, heroin addicts, and H, hemophiliacs. People suffering from disease of the blood known as the hemophilia. They were noted to be dying or normally benign opportunistic infections. Typical cause of the death of people infected by HIV virus is the development of the opportunistic infection. You remember maybe that opportunistic infection is the infection caused by usually low pathogenic microbes, bacteria, viruses, or fungi or parasites. In the normal people with the normal state of the immune system, these parasites can cause the clinically manifested disease. But in immunosuppressed people, these microbes start to colonize human tissues effectively. They bypass the weakened immune response and cause the severe symptoms of the disease that are usually lethal for the people. Brief history of discovery. On May 1983, Luc Montagnier in the Paris, that worked in the Institute of the Pasteur, published first known description of unknown virus named Lyme. This is LAI, this is the name of the virus formulated by Montagnier. 1 year later, in 1984, American scientist Robert Hallow with the co-workers that worked at the National Institute of the Health of the United States reported the isolation of the human immunodeficiency virus, that is named the immunodeficiency virus type 1, from patients with lymphadenopathy and AIDS. AIDS, this is a disease caused by opportunistic agents in immune-suppressed people. Each group of scientists, group of the Montagnier and group of the Galland, gave their viruses different names. Later, committee of the international experts named virus discovered by both these groups of the scientists as the HIV, human immunodeficiency virus. Structure of retroviruses. Ladies and gentlemen, 1 second, please. Wait, please.
никто не отвечает.
Это что, один человек?
И один человек отметился, что...
Ты знаешь, что это такое? Для одного человека читать лекцию.
Сейчас еще
Он есть. Разрешите я тогда перейду. Что я должен нажать, чтобы...
на PowerPoint.
Вот сюда?
Да.
Понятно. Excuse me, I will continue. Structure of retroviruses. No, in this figure you see structure of the one of the human immunodeficiency virus type 1. But this is a common characteristic of all the retroviruses. Structural characteristics. Retroviruses have spherical envelope vision, or spherical envelope virus particle, with a diameter from 80 to 120 nanometers. This is an intermediate size virus. This virus have the what HIV virus. Typical core, shown in the yellow central cylindrical part of the virus. This is a core or capsid of so-called D type. This is a cylindrical capsid surrounded by the envelope. Envelope shown green and consists of the lipids and integrated to these lipids surface glycoproteins. Human immunodeficiency virus contains in the envelope two types, two proteins, glycoproteins. They are known GP41, this is a transmembrane molecule, and GP120, external oval part of the transmembrane molecule. Both these glycoproteins, GP41 and GP120, they form the attachment protein of the virus that attaches this virus to the human cells sensitive to these viruses. First of all, T helper cells and macrophages. By the way, glycoproteins coating the virus associated to the envelope of this virus. This is these glycoproteins are the major immunological markers of the human immunodeficiency viruses. The core morphology is used to classify these viruses for different groups. Note this is of the D type. In the photograph in the right part you see of the slide you see truncated cylindrical core. This core in the theoretical scheme is shown yellow. In the electron microscope looks like the black cuneiform structure. The genome of retroviruses, including HIV. Genome of retroviruses is single-stranded, linear, positive sense RNA with a size from 9 to 10 kilobase pairs. The unique characteristic of the retrovirus genome is that the genome is diploid. What you see, one and second RNA molecules, two RNA molecules per one virus particle. Each RNA molecule shown here is identical to one other one. Presence of the diploid genome is the distinctive characteristic of the retroviruses. The next distinctive characteristic. The virus core contains molecule of the reverse transcriptase, or RT. Every RNA molecule of the diploid genome contains one reverse transcriptase molecule attached to this RNA. Also, every RNA molecule contacts to the second enzyme that is named integrase, virus integrase enzyme. Every RNA molecule of the virus genome is attached to the cellular transport RNA. Two molecules of the cellular human transport RNA to one RNA molecule of the virus genome. This RNA serves as the primers for the reverse transcription, for the initiation of this synthetic process. Viruses, HIV virus and other viruses, have also virus-encoded protease. This enzyme occurs in the virus particle and suffers the molecules that may cleave, so-called polyproteins, in the period of the translation of the virus proteins. In this slide, you see theoretical structure of the linear RNA genome of the retrovirus. You see that different genes of the genome are flanked by the two identical elements that occur at every end of the RNA genome. These terminal parts of the genome are named long terminal repeats. Long terminal repeat number 1 and long terminal repeat number 2. Long terminal repeats, the sequences of the nucleotides, contain so-called promoters, enhancers, and other gene sequences that are used for binding various cellular transcription factors to the genome of the retrovirus. Here you see the same theoretical structure, but you see that virus HIV genome RNA contains different blocks of genes. The major blocks of the genes is GAG, or GAG, POL, second block, and ENF. ENF, this is a brief form of the envelope. ENF genes that occur in the ENF genetic fragment, they code for glycoproteins, GP41 and GP120 of the envelope of the virus. Every of these three large blocks codes for polyproteins. Polyproteins shown here the white, orange large molecules. It means, for example, genetic segment, when it's translated, maintains synthesis of the POL polyproteins. POL, this is a brief form of the word polymerase. These polyproteins contain at least 4 different enzymes that are encoded by the virus genome. One of the proteins encoded in the POL genetic block is P10. It means protein 10, 10,000 intons, or protease. In the lower part of the scheme, you see maybe smallest, no, not smallest, but one of the small molecules, P10. This P10 molecule is the protease that cleaves on POL polyprotein, releasing functional proteins, releasing itself. The P10 cuts on molecule from the polyprotein, POL polyprotein, releases from polyprotein P6CC P51, this is a reverse transcriptase, releases P15, this is a RNase, and releases P32, integrase. We told you that reverse transcriptase, integrase, and polymerase, they occur in the virion particle. P10 also cleaves other polyproteins, GAG polyprotein and ENF polyprotein. From GAG polyproteins are released proteins that form capsid P17 and P24, and that have nuclear characteristics. It means code each the molecule of RNA. And we told you that GP160 polyprotein, when it's cleaved, releases from itself GP120 and GP41, these two surface proteins. So you know that retroviruses have GAG, POL, and ENF genes, or genetic blocks. GAG is translated in the group-specific antigen. It means that GAG proteins are immunologically conserved. An antibody against GAG proteins of the human immunodeficiency virus type 1 isolated in the 1985 here, immunologically is identical to the analogous protein isolated from the virus in the 2019th year, for example. GAG specific antigen, this block codes for P24, shown yellow. This is a P24 that forms a cylindrical core. That's one of the most important and distinctive elements of the virus capsid. Also, among GAG proteins, you can find P17 or matrix protein. This matrix protein forms spherical shell surrounded from outside by the lipids of the envelope. Also, P7 is encoded and P6 is encoded by GAG genes, and these proteins P7, P6, and P9, they code every molecule of the genomic RNA, forming the individual tube surrounding RNA that occurs inside. The next genetic block, and correspondingly genes and correspondingly proteins. POL, polymerase. POL genetic fragment codes for P10. This is a HIV protease. P66, P51. This is reverse transcriptase. And P32, integrase. And one molecule more, P15, that is named RNase, that may destroy ribonucleic acid. And the last genetic block and block of the proteins is the ENF block. ENF proteins, or envelope proteins, they are two glycoproteins, GP proteins, means glycoproteins, that form the attachment protein of the attachment protein complex of the HIV virus, exposed to the outside of the virus and serve as the attachment protein of the virus. One element of this ENF proteins is GP41 that forms transmembrane stalk part of the envelope attachment complex of the HIV, and GP120. Both GP41 and GP120, they have the lollipop structure. Genome of the retroviruses, including human deficiency virus type 1, except for structural GAG, POL, and ENF proteins, have genes that code for accessory or regulatory proteins of the virus. They are TAT genes, REF genes, NEF genes, and corresponding protein, VIF, VPU, and VPR No, for example, TAT. TAT gene codes for protein P14 that may transactivate of the virus and cellular genes for transcription. It means TAT gene is a transactivator that regulates transcription of both some human and the virus genes. REF gene codes for P19 that is responsible for regulation of RNA splicing and promotion of the export of the virus products from nucleus to the cytoplasm, export of RNA, first of all. NEF gene calls for P27. This P27 reduces producing of the chemokines by the virus-infected macrophages. NEF protein may activate resting T cell activation that is associated to the progression of the acquired immunodeficiency syndrome. It means NEF may stimulate finishing of the non-symptomatic period and beginning of the AIDS. VIF gene calls for P23, this is a VIF protein, that promotes the virion assembly. It means it's important in the building of the progeny virus particle. VPU gene calls for VPU P15 protein that facilitates release of the virus and the decrease of cell surface CD4. And VPR gene calls for P15, VPR P15, that is possible for transport of the complementary DNA to nucleus and the arresting of cell growth. All these genes in the scheme that you see here are shown as the short genetic components of the genome of virus that are localized mostly in the central part of the genome. What you see here, where is located VPR, REF, TAT, VIF, VPU genes that are mostly situated between PO and ENF genetic blocks. Proteins that are encoded by these accessory genes, NEF, VIF, TAT, they may bind to the specific sites in the long terminal repeats, switching on some of the processes that are functions of the proteins that are encoded by these accessory or regulatory HIV proteins. Replication of the retroviruses. In this scheme you see the low part of the scheme, fragment of the envelope of the retrovirus contain 1, 2, 3, 4 lollipop structures consisting from GP41, the transmembrane protein, and GP120, the external spherical element of this spike. Virus spikes contact to the receptors of the cells. Mostly these cells are CD4 positive T helper cells and macrophages. So not only macrophages, but cells of the macrophage line, the monocytes, dendritic cells, in the process of the interaction of the virus is not shown completely, but here is shown only fragment of the envelope, some lipids, with the spike consisting of the GP120, GP41. GP41, this scheme shown in blue. GP120 consists of 4 GP120 molecules that are associated to GP41. So, HIV binds to the cellular CD4 receptors. CD4 receptor, cell membrane of host cell is shown as a lipid layer below. In this lipid layer are integrated CD4 molecules and so-called blue co-receptors. GP120, first of all, finds and binds to the CD4 receptor of the T helper cells and macrophages. After this binding, configuration of the virus spike is changed, and other elements of the GP120 binds additionally to the C co-receptors shown in blue. These co-receptors are CCR5 on the macrophage and CXCR4 on the surface of the T helper cell. Co-receptors are the transmembrane G protein coupled chemokine receptors with CCR, when saying about receptor for macrophage. After binding of the G120 to CD4, internal parts of the 120 domain comes open for contact to the co-receptors, and when both CD4 and co-receptors bind to the GP120, configuration of the spike structure is changed. GP120 configuration is changed, and GP41 elements becomes exposed to the surface of the lipid membrane of the human cell. These GP41 components, they becomes integrated to the lipid of the human cell, and lipid membrane of human cell is attracted to the lipid membrane of the virus envelope. As a result, virus envelope fuses to the cellular membrane, and internal parts of the HIV becomes injected to the cytosol of the newly infected cell. Replication cycle of the retrovirus is shown in this scheme. Now take a look to the upper part of the slide. You see in the square zone the structure of the matured HIV virus. On the left, you see how this virus attaches itself to the cellular cytoplasmic membrane via binding of GP120 to the CD4 and binding to this molecule of the CCR5 or CXCR co-receptors that mediate fusion of the virus particle to the cell with the release of the core structure from the virus particle to the cytoplasm. In the cytoplasm, virus core is very quickly breaks down, and nucleic acid, RNA, is undergone to the transported to the nucleus. Nucleus is shown here in the blue zone in the lower part of the slide. RNA in the core before coming to the nucleoplasm is going to the reverse transcription. Reverse transcription creates DNA intermediate after the reverse transcription and synthesis of this complementary DNA as the template of the virus RNA genome. With this DNA is transported to the pores in the nuclear membrane, and without P24 that is shown green here, DNA is nucleated to the nucleus, and in the nucleus, as a result of function of the integrase, DNA intermediate copy of the virus genome is integrated to the genome of the patient. And after this integration, DNA genome of the virus occurs as the part of the genome of the human cell. This DNA is transcribed, and a group of different transcripts are formed. So here these RNA transcripts are shown as the molecules that are transported from the nucleus to the cytosol. You see here green spliced messenger ribonucleic acids synthesized on the basis on the template of the virus DNA. These mRNAs are involved in the translation, and each of these fragments calls for a certain polyprotein. As a result, in the Golgi period is synthesized GP160. This is a precursor of the mature GP120 and GP41. GP160 appears on the internal membrane of the structures of the endoplasmic reticulum and Golgi period, and after that, with these chambers of the endoplasmic reticulum and Golgi period, this GP160 are transported to the surface of the host cell. At the same time, in the other parts of the cytoplasm are synthesized other proteins, regulatory proteins, excessive, for example, NEF and VPU, that may block some synthetic processes in the cell. For example, NEF protein of the virus is a blockator of synthesis of CD4 molecule of the infected cell. MHC1 molecules are also inhibited. Other proteins, non-enable but pol and GAG-pol proteins, are synthesized outside of the Golgi period, the ribosomes in the cytoplasm of the cell, and polyproteins, GAG polyprotein and pol polyproteins are transported to the cytoplasmic membrane of the cell, modified by glycoproteins GP160, and below this area, when this human glycoprotein is situated, are accumulated GAG polyproteins and pol polyproteins, and during the accumulation of these proteins in this place, BAT is formed, and finally BAT, when it's formed, is released from the virus, and you see the non-matured virus structure. Why non-matured? Because surface GP molecules are named GP160, Later, they are cleaved by reverse transcriptase, by protease that occur in the virus particles. Protease cleaves polyproteins, and after that, completely matured virus proteins functionally active are synthesized, and after some certain period of time, this provirus is transformed to the completely matured virus. In the completely matured virus, cylindrical core is formed. Usually when cylindrical core is appeared, it means that certain this virus particle is completely matured. It means maturation of the virus particle is continued after release of this particle by the binding from the host cell. And the virus particle completely matured after the certain time when virus protease cleaves all the polyproteins. Now here you see translation and self-assembly. No, translation, you know, this is a synthesis of proteins. In this scheme, you see how in the Golgi heparin appears 120 and 140, GP120 and GP41. GP120 and GP41 as a result of cleavage of the nth polyprotein by the virus protease. Virus protease initially is synthesized as a part of the GAG polyprotein. What here is shown GAG polyprotein consisting of the elements of different shape and different color. With purple component, this is a protease. This protease cuts itself from this polyprotein, and after that releases other functional proteins from the structure of the polyproteins. But in this process, during this process, before finishing of this process, various polyproteins, GAG polyproteins and POLQ polyproteins bind to the newly synthesized genomic RNA molecules, surround these molecules, in this moment react to the transmembrane parts of the GP41 newly synthesized, form buds, but it's released, and during all these episodes, various protease step by step cleaves polyproteins, transforming them to the functionally active virus proteins. Process is finished after binding of the virus particle. The result, completely matured virus is produced. No, self-assembly, you understand, is the process of combining of all the elements of the variants in one complex. GP120 and 41 appear in the membrane of the cell. Transmembrane portions of GP41 interact to the P17 with purple elements that directly bind to the GP41 and stick part of the cell membrane to the GP120, GP41 and P17. But P17 before cleavage is bound to the P24. P24 here is shown by green color. P24 shaded initially to the P7, P9, P6. RNA binds to this proteins before their cleaving, become surrounded by small gag molecules, and parts of these complexes are pole genes also, including protease, that during the episodes of from the beginning of bud to the formation of the bud and the releasing of this bud, during all these periods, various partners continues permanently cleave new polyproteins, step by step reducing completely mature proteins. After the maturation, they change their configuration and finally occupy the zones that are typical for these proteins in the completely matured virus particle. Now, virus particles are released from cells by the body. What you see, these buds here, the electron microphotograph. In the slide that you see here, you see the left part, unmatured virus particle, and the right part, matured virus particle. You see that in matured virus particle, virus proteins, first of all, GAG proteins and pole proteins occupy zone surrounding region containing RNA, but they don't form the cylindrical core. After maturation, different proteins, after their cleaving, releasing separation from other proteins by the virus protease, occupy so-called correct areas of the virus particle, and the virus particle appears completely matured. That is shown in the right part of the slide. What is the brief extract from what is this slide? Extract is virus proteins remains active after budding, and in the early period, this virus particle after budding is non-infectious, because this particle can penetrate membrane of the cell, it can't establish infection. But after permanent functioning of the virus protease, step by step, this precursor particle becomes mature to the absolutely functionally active virus particle. It means virus protease works intensively after budding of the virus particle. Except for producing of the virus particles, HIV may maintain process of the syncytia formation. Syncytia formation, this is a fusing of the virus-infected cells to the giant cells that consist of the many nuclear elements of many cells, and size of this syncytia may be 100 times larger than size of the non-infected cell. Non-infected cells are shown as the small, very small ovals shown here. Syncytia shown as the very large, spherical, but not obviously spherical, giant structures that are a result of the fusion of many cells maintain virus replication inside. Genetic divergence. The genetic divergence, this is a process typical for many viruses, not only for HIV. Usually divergence is intensively studied for viruses that very quickly produce immunologically different variants. For example, typical for influenza viruses and for retroviruses. A divergent population of viral genomes comprises a spectrum of non-identical but related HIV genomes. Of these related HIV genomes, new genomes appear during the life of a single patient, and divergent strains of the virus may be isolated from one person in the different periods of the development of the infection. You remember that subfamily that involves HIV viruses is named lentivirinae. Means viruses causing lenti process, long time process, that without treatment may last for 4, 5, 6 years. This is a period of survival of patient. But during these years, from patient may be isolated in different periods, different divergent variants, genetically changed. Why retroviruses may produce these new genetic variants, immunologically different variants? This is due to the function of the reverse transcriptase. Reverse transcriptase produces during the reverse transcription many mistakes during one synthetic cycle, and DNA copies of the RNA genomes contain many mutations caused by reverse transcriptase. And if these mutations are not lethal for newly formed viruses, viruses survive having with these changed fragments of the genome that explain variability. The most variable part of the HIV, the glycoproteins, and from them GP120. But GP120 usually contains a large number of the mutations generated by reverse transcriptase than other genes. Parts of the molecule of GP120 that usually undergo into this mutation process are named variable regions. There are many variable regions now described, have been described, and the most variable is so-called variable number 3 region, is the that is considered the major antigenic determinant of the HIV. With this variability and mistake-prone function of the reverse transcriptase that generates these mutations is a major cause of the problems in the development of the anti-HIV vaccines. Many variants of vaccines have been produced and described and studied until now, but now no one vaccine was considered effective, because with this genetic divergence and immunological variability of the HIV makes all these vaccines non-effective, because they generate immunity against certain variants. During synthesis of these producing vaccines, new immunological variants appear, new antigenic variants appear, and the vaccine becomes capable of producing immunity, effective immunity against HIV. Pathogenesis of the HIV infection. Pathogenesis. HIV has the tropisms for CD4 expressing T cells and CD4 receptor expressing macrophages. So T-tropic strains and M-tropic strains of the HIV. HIV induces immunosuppression. deep immunosuppression, demonstrating the appearance of the opportunistic infections and tumors, is named AIDS, acquired immunodeficiency syndrome. We have to remember that AIDS is the syndrome that appears after years after beginning of the infection. It means newly infected people during 2 or 3 years, they don't have AIDS. They have HIV infection, but if in these patients immune system is not destroyed, they don't demonstrate appearance of tumors and opportunistic infections, and don't die, don't demonstrate symptoms. When we say about AIDS, this is the last period of the HIV infection with a severe course of this infection that lasts usually not more than 1 year. About 1 year after appearance of signs of the AIDS, patients usually die. First cells, immune cells that contact with the HIV and recognize HIV are the antigen-presenting cells of the macrophage line. First of all, dendritic cells, they are shown green here. Dendritic cells internalize HIV because recognize HIV pumps, pathogen-sweet molecular proteins, by the pattern-recognizing receptors, including talc-like receptors. Dendritic cells internalize HIV after this recognition and migrate to the lymph nodes, keeping the HIV virus genome inside. But here is the nucleus and genes of the genome of the retrovirus. And dendritic cells serve as the transport systems that later transfer virus to the lymph nodes and play an important role in the infected of the CD4 T cells. It means first cells infected and antigen present. After that, antigen presenting cells infected by HIV, they present antigens of the HIV to the T cells and infect these cells. Really transfer virus to these cells and the CD4 cells becomes infected. Second, immature Langerhans cells. Langerhans cells, they are shown as the purple that occur in the epithelial line. They express protein that is named langerin. But here this langerin is shown as the purple molecule and green molecule inside. And these immature Langerhans cells promote viral degradation in the so-called Birbeck granules. But they also may transport virus genome and viruses to the other cells, including macrophages. Macrophages that are localized in the submucous epithelial, submucous tissues, they take up HIV via mannose receptors. Mannose, the pattern recognizing, one of the pattern recognizing receptors of the macrophages. Macrophages accumulate viruses inside. They usually become infected by stem cells and serve macrophage as the most important reservoir of the HIV in the human organism in the early periods of the infection. And first strains of the HIV virus that appear in the tissues of the human being freshly infected, they are macrophage-associated HIV strains. Later, during process of the divergence, these macrophage HIV strains becomes transformed to the T cell strains that infect CD4 T cells. So all three innate immunity cells shown here, dendritic cells, lung cells, and macrophages, express toll-like receptors, the receptors that also react to the virus pathogen-associated molecular patterns. And firstly, virus accumulated in the human body in the cells, the macrophage line, what the cells that are shown in this slide. So initial infection of macrophages with the M-tropic HIV strains directly towards CD4 and CCR5 receptors of macrophages. This is the first step of the occupation of human body by the HIV. Macrophage lineage cells becomes persistently infected and surface the virus reservoir. These cells are circulated macrophages, microglial cells, alveolar macrophages, dendritic cells, and other cells of the monocyte macrophage line. They spread the virus and contribute to the beginning of the clinical manifestation of the infection. Later appear cell-tropic HIV strains, and appearance of these strains serve as the marker of the progression of the disease to the AIDS. In this slide, you see CD4, CXCR4 chemokine receptors contain THAV. Well, this is on the left part you see T helper cell, normal. In the right you see HIV infected T helper cell. From that many virus particles, but you see what these spherical elements of the cell, they are bodied virus particles. In the pathogenesis of HIV infection, the virus cytopathic effect play an important role. This is a killing of the cells by following mechanisms. Mechanisms of the killing of the cells by the HIV are accumulation of non-integrated cellular DNA copies in the nucleus and cytoplasm of the infected cells. This accumulation of the DNA virus intermediate by itself may cause degradation of cell. Second, cytopathic effect of viruses is realized via increased permeability of the plasma membrane. Plasma membrane becomes penetrated by virus like a proteins, and this makes this plasma membrane not permeable for different substances and structures, and the plasma membrane may be ruptured. Result of the modification of plasma membrane is the syncytia formation, fusion of many cells, and the complex consisting from many cells. And finally, of course, all these processes, including syncytia formation, causes apoptosis of infected cells, and they die. So, in the pathogenesis of HIV, two lines of cells mostly are infected. Macrophages, shown on the line, this sequence ferment is shown at the right part of the scheme, and CD4 T helper cells. What is happened when macrophage line of cells is infected. Initially, persistent infection of macrophages happen, and dendritic cells and monocytes, it means cells of the macrophage line. After that, brain diseases are developed due to the damage of glial cells, and the neurophysiological manifestations, symptoms appear. Mental sphere of the patient is changed. When T line of cells is infected, initially also persistent infection is developed. After that, productive infection with the producing of high virus load appears. After immunosuppression is developed with the depletion of CD4 line of cells, and opportunistic infections and tumors, neoplasms, the neoplasms are developed. Opportunistic infections are developed. In this slide, you see result of the computer tomography of the brain of the patient in the different periods of the disease development. In the A photograph, you see normal figuration of elements of the brain. B photograph demonstrates B 4 months later, when the mental manifestation is developed. You see loss of the brain cells, loss of the brain material that is shown by the red arrow. Profound loss of brain substances here. The cause of the HIV infection. Cause of HIV infection parallels the CD40 cell number and the virus amount in the blood. HIV infection is treated with burst of virus producing and viremia in the beginning of the disease. In this figure, you see very narrow and large peak, this number of the virus particles, and after in the beginning of the disease is the fresh disease, first stage of this disease. Later number of the viruses for the long time becomes lowered, and after in the last period of the disease, that is named as the AIDS, number of virus particle, concentration of them is increased also, virus load is increased. So, burst of the virus producing viremia in the beginning of the disease. Occurrence of the mononucleosis-like symptoms, usually associated with this first and short period of the HIV infection. Later, virus level in the blood is decreased for a long time, but virus replication continues in the lymph nodes. The clinically latent period occurs. This latent clinical period what is characterized by increase of level of the antibodies, anti-HIV antibodies. This is a green curve. And characterized by the period of the low virus load in the tissues. During development of this disease that lasts 4 or 5 years, level of CD4 cells, shown red in this scheme, is decreases. And when this level becomes very low, it means about 200 cells, CD4 cells per 1 microliter of the blood, immunosuppression becomes very severe, and in this period appear symptoms of the opportunistic infections. This is pre-AIDS, highly developed opportunistic infections, and tumors, they characterize AIDS. AIDS, this is the last period of the disease, associated to what period from 60 months to the 80 months. But the patient, disease of that is shown here. Reducing of the number of CD4 cells during disease development is explained by the cytolysis caused by virus, activity of T killer cells, CD8 cells that recognize virus-infected CD4 cells and kill them. And the third cause of the depletion of the CD4 cells population is the natural terminal differentiation of T cells in the presence of very high concentration of the virus particles. AIDS period is shown here by this red symbol AIDS. The last period when virus load becomes high again and number of CD4 cells is very low. So this is some words about history of the development, history of the discovery of the HIV. The theory explain appearance of the HIV in the human population, most common today, declared that HIV is the result of the evolution of the monkey retroviruses in the chimpanzees and other species of the monkeys that live in the Africa. Hunting for the monkeys causes the transfer of the monkey retroviruses to humans, evaluating the high variability of these viruses. Monkey virus strains after adaptation for humans was the appearance of the virus that is known now as the HIV-1. In the North Africa now is a typical HIV-2 virus that is similar to the simian monkey viruses, typical for the smooth manga base in the Western Africa. HIV is spread higher than HIV-2 and is considered as the more adapted for the human hosts variant of the HIV-2 virus, and both HIV-2 and HIV-1 now cause pandemic that is known as human immune deficiency virus pandemic. What the factors facilitate spread of the virus? HIV is present in the blood, semen, and vaginal secretions. So, virus is spread through sexual contact, through the contaminated blood products, other types of grafts that are used in the transplantation. And also HIV may be transmitted perinatally to the newborns from the infected mothers. Population at high risk are understandable. Clinical syndromes typical for HIV. Beginning of the infection. Initial HIV infection is characterized by the influenza-like symptoms or infectious mononucleosis-like symptoms. Increasing of the body temperature for the 1, 2, or 3 weeks. So throat, cough, and sneezing. After these 2-3 weeks of the clinically manifested early period, symptoms disappear. In this period, rash or aseptic meningitis may occur. They are very rare symptoms, skin rash or aseptic meningitis, but they have been described. that may appear during the first 3 months after beginning of the disease. The second period, initial symptoms period, what first, is followed by a period of asymptomatic infection. This is a rather long period of asymptomatic infection. Asymptomatic or persistent generalized lymphadenopathy. In some people appear swelling of the lymph nodes in the different anatomic regions of the body without other symptoms. This situation is named as the persistent lymphadenopathy. Then, period number 3, period of the development of the opportunistic infections that resulted of a deterioration of the immune response that correlates to the lowering of the number of CD4 cells. Opportunistic infections usually appear in people that have less than 450 CD4 cells per 1 microliter of the blood. Less than 450 CD4 cells. When number of the CD4 cells is low, down to the about 200 CD4 cells per 1 microliter, so-called full-blown AIDS is developed. That's shown by what's zone number 4. Full-blown AIDS is characterized by the so-called HIV wasting syndrome, body weight loss and diarrhea for more than 1 month. Also, Kaposh's sarcoma is developed. This is a tumor caused by herpes viruses. This is an indicator disease for HIV patients. Third typical disease is pneumocystic carinii pneumonia. Pneumonia caused the protozoa named as pneumocystic carinii. This is a major sign of AIDS. An infection caused by Mycobacterium avium intracellular complex and the cytomegalovirus infection. What they are opportunistic infections that characterize AIDS period and the HIV infection of the patient. Here you see the wasting syndrome. Patient with the wasting syndrome associated with HIV, lipidystrophy AIDS. In AIDS is happened. Lipidystrophy, it means loss of the fat. Fungal opportunistic infections develop, for example, aspergillosis. You see X-ray examination with a large area in the lung, ball-like, that is filled with the aspergillus fungal organisms. This is so-called aspergilloma. Candidiasis. Candidiasis is caused by Candida albicans and other species of Candida. This typically causes disease in mucous membranes, but candidiasis also may cause systemic spread with the formation of sepsis. Oral candidiosis is shown here. You see on the pellet, on the surface of pellet, white colonies of the Candida albicans. Candidiasis of esophagus is typical for these patients. Candidiasis of trachea, bronchi, and lungs. Here you see disseminated cutaneous sporotrichosis. Sporotrichonki, this is an opportunistic organism causing damage of internal organs and skin. Kaposh's sarcoma. This is a tumor. Tumor cells are cells of the blood vessels. Practically, you see formation of tissues consisting of the vessels. Due to this, bleedings are very typical for patients with Kaposi sarcoma. Kaposi sarcoma may occur in different parts of the human body. What here you see, mouth, where soft palate is affected. You see the leg appearance of this red purple tumor, and the skin rashes that also consist of the tumor cells on the face. Oral leukoplakia. This is also opportunistic infection process caused by other viruses. Laboratory diagnosis. Laboratory diagnosis of the HIV infection consists of many elements. Finding of P24, but in this figure, P24 level at different periods of disease shown by the yellow curve. P24 level, you remember that P24, this is protein that waits for 24,000 Dalton. That is a major element of the virus capsid. This P24 level may be detected by ELISA, enzyme link immunosorbitase, and correlates to the virus load. Virus culture also is available, but routinely culture is not used for diagnosis in the clinical conditions because it's very expensive and long-lasting. Reverse transcriptase level may be determined by the special chemical tests. Viral RNA detection is very important. Viral RNA detection may be done via the reverse transcriptase polymerase chain reaction. But directly you may count number of the viral RNA molecules in the cubic milliliter of the blood or other human body, and in this way we can measure the so-called virus load, the concentration of virus genomes in the human tissues. And of course, serological tests. Serological tests are directed for finding of the antivirus antibodies. Usually, tests for serology are ELISA for antibodies, Western blot, or radioimmunoassay or immunofluorescence also may be done. But usually this is ELISA and Western blot. The result of the serology testing is shown by this blue curve, characterizing the amount of the antivirus immunoglobulins that occur in different periods of the disease. And of course, counting of the CD4 cells is important as a characteristic of the state of the immune system of the patient. Serology. Routine screening of the patient's blood includes use of the ELISA. But ELISA or latex agglutination test, usually ELISA. ELISA can yield so-called false positive results. And to bypass all this limitation of ELISA, Western blot is usually used as a method of the confirming of the positive results, demonstration by the ELISA. In the photograph here, you see 10 stripes with the virus proteins that are fixed in the different parts of the stripes. These stripes are sold by the patient's serum after incubation and washing out of the excess of the serum. Anti-human antibody reagents, anti-human antibody usually labeled, used to visualize zones where virus proteins interact with the human antibodies. Most important findings to confirm positive diagnosis. This is a demonstration of the patient's antibodies that react to the GP160, the upper zone. GP160, this is a unmatured glycoprotein that may occur in the virus-infected cells. The second, GP120, this is a mature attachment protein. GP41, also attachment protein. Exception for that, important but less important, is the finding of P23, P24 with the low zone, P31 and other, P17 and other proteins that are present in viruses may generate antibody synthesis. Now here you see culture. Culture is used now mostly in the study of the effectiveness of the antivirus preparations and study of the effectiveness of antivirus vaccines. In the lower part of the slide you see that very important is the counting the absolute number of CD4 and of the ratio of CD4 to CD8. CD4 to CD8 ratio usually in normal people is more than 1. In the patient with HIV infection, this ratio is less than 1. Antiretrovirus therapy. Antiretrovirus therapy had been started in the 1987 when first antiretrovirus preparation have been discovered and introduced in the therapy. This drug was named zidovudin. Now zidovudin is considered the first drug in the group of so-called nucleoside analogs, retrovirus nucleoside analogs. So, treatment of the HIV mostly is performed using following preparations: nucleoside analog reverse transcriptase inhibitors, nucleoside analog reverse transcriptase inhibitors. But zidovudin or zidetimidine that we characterized before is the first in this group. Second group of antiretrovirus preparations, non-nucleoside reverse transcriptase inhibitors. Third, protease inhibitors. You remember that protease is responsible for maturation of the virus particles. Here you see a list of the nucleoside analog reverse transcriptase inhibitors. First is zidovudin, second zalcitabine, next didanazine, stavudin, lamivudin, abacavir. These drugs are rather toxic, and due to this, they are not used now in the monotherapy. They usually combine to the other antiretrovirus drugs that allow us to use a lower concentration of, for example, zidovudin in the treatment of the patients. The next group of the drugs are the protease inhibitors. Saquinavir, ritonavir, indinavir, nelfinavir, amprinavir are examples of the protease inhibitors. The next group, non-nucleoside reverse transcriptase inhibitors. These substances change the properties of the reverse transcription active site, reducing activity of this enzyme. Non-nucleoside reverse transcriptase inhibitors are nevirapine, delaverdin, efavirenz. Now combination or multi-drug therapy is used, where you treat your patient using combination of the anti-HIV drugs, where in the combination you include at least 3 drugs that are members of at least 2 different groups of the anti-HIV drugs. For example, what indinavir may be associated with azidotimidine and lamivudin. Azidotimidine and lamivudin are nucleoside reverse transcriptase inhibitors. Indinavir is an inhibitor of the protease. What lower you see 2 combinations more. So what you have to remember, a cocktail of several antiviral drugs with different mechanisms of the action are recommended now to be used in the treatment. This treatment by the complexes of anti-HIV preparations that are members of different groups of the drugs now is named as the highly active antiretrovirus therapy, HAART. Also, this highly active antiretrovirus therapy may be named as the combination antiretrovirus therapy. Now you see inhibitors of the initial steps of the infecting of cells. You see here that, for example, fusion inhibitors, this is a group of the drugs, fusion inhibitors of the efavirenz, may inhibit fusion of the virus particles to the membrane of the membrane of the human cell. Fifth group, co-receptor antagonists. You know that binding of the GP120 to CD4 receptor is not enough to realize penetration of virus particle to the human cell. The next step would be the reaction of the GB120 to the CCR5 co-receptor, for example. Well, this binding to the co-receptors is inhibited by the such preparations as the maraviroc or Krivorutchenko. They are preparations that appeared during the last 5 years, and they are approved for use, but experience of their use is not very long. No, this is a maraviroc. Integrase inhibitors are raltegravir, elvitegravir, dolutegravir, and cabotegravir. These inhibitors of integrase interact with the virus integrase enzyme, preventing it from the inserting the viral DNA to the human genome. Raltegravir, for example. Attachment inhibitors, Rucobia, for example. They are substances that prevent attaching of the GP120 to the CD4 molecule. And the fostemsavir, for example, is one of these attachment inhibitors. It means attachment inhibitors, integrase inhibitors, chemokine receptors antagonists, co-receptor antagonists, fusion inhibitors. These drugs are comparatively newer drugs that also are used now in the treatment of the patients, but classical therapy, this is an antivirus therapy, consisting of the use of the reverse transcriptase inhibitors, protease inhibitors, and nucleoside and mononucleoside reverse transcriptase inhibitors. What is this scheme? You see different examples of the therapies that are used now. No, first, maraviroc. This is a co-receptor antagonist preventing binding of GP120 to the co-receptors. Efavirenz prevents fusion of the virus to the cytoplasm. Then raltegravir integrase inhibitor that prevents integration of virus DNA to the human. First line preparation, this is zidovudine and other analogs of the reverse transcriptase inhibitors that prevent reverse transcription. It means synthesis of the virus complementary DNA on the basis of the RNA of the virion. And of course, protease inhibitors, for example, indinavir. Protease inhibitors start the prevention of function of protease before budding, during the budding, and after budding. Protease inhibitors becomes integrated not only to cell but to the newly formed virus particle, where do they prevent maturation of virus particle to the infectious matured virus particle. Other parts of the lecture are understandable. One word about perspectives of the vaccines. Worldwide attempts of the production of the vaccines against HIV, history of these attempts occupies at least about 40 years. During these 40 years, different variants of the experimental vaccines have been developed. But due to the very high genetic and immunological variability of the virus, HIV virus, no one vaccine that now is available. It means retroviruses, they are viruses that are very difficult to be controlled by the vaccines made on the basis of principles that are known now. It means that possibly vaccine against HIV would be impossible to be produced if progress in this plane would be happen. Most possibly that this theoretical progress may be associated to the finding of new principles of creating of the vaccines. But until now, these new principles are not known. It means HIV, this is a virus that can be controlled by vaccination, and perspectives in this plane are unclear. It means prevention of this disease may be based only to the following to the rules of the controlling of the HIV infection in the medical facilities and the life, for example, safe sex, using of the disposable instruments, hand washing, glove wearing, and so on. It means these principles are very common with the principles of prevention of the coronavirus infection. What else? You saw that now many drugs are approved to be used in the treatment of the HIV. And really, intensive antiretrovirus therapy may prolong the life of the HIV-infected person to the many years. Now, exams of the maintenance of the infected pit during more than 10 or more than 15 years using correct change of the preparations. These exams are very common. It means you can use therapeutic drugs now mostly as the drugs for therapy and drugs for prevention of HIV. Now this is a finishing of my lecture. Goodbye.